Hi, welcome back. What I want to do in this video is really explore the Tca cycle. And what we saw in the last videos, we looked at the pyruvate dehydrogenase complex. And what we saw is that in that pathway, or that enzyme, we produced acetyl-CoA. And in this video, we're going to look at all the reactions in the Tca cycle up to the production of succinyl-CoA, and then we'll finish the pathway in another video. Now, one thing I will tell you is I'm going to draw this in a linear fashion, but just know that it is a cycle. And it's important to distinguish between a pathway and a cycle. A pathway is a linear reaction scheme, meaning that it doesn't, the original substrate doesn't cycle back to produce, essentially produce it again. A cycle, it ends up regenerating what you started with. So in this pathway, we're going to start with citrate, essentially, and we're going to regenerate citrate. So therefore, it's a cycle, although I am going to draw it in a linear fashion. Now, the very first enzyme in this pathway is basically citrate synthase. And one thing I'll go ahead and tell you, and this is important, is that citrate synthase is the first allosteric enzyme of the Tca cycle. So citrate synthase is allosteric. And essentially what's going to happen with citrate synthase is water is going to come in, and it's going to remove the CoA, and ultimately what you're going to generate is citrate, or sometimes I pronounce it citrate. And citrate is a 6-carbon molecule. Citrate is a 6-carbon molecule, and it looks like this. It looks like this. So there's citrate. So notice that citrate not only is 6 carbons, but citrate has 3 carboxylates. Here's the first one, here's the second one, and here's the third one. So citrate has 3 carboxylates, and that's actually the name of where the Tca cycle comes from. It's the tricarboxylic acid cycle. So the tricarboxylic acid, or the tricarboxylate, refers to citrate. And actually, you can also, as we'll see in the next reaction, you can also refer to it you can refer to the tricarboxylate as isocitrate. Isocitrate also follows the same rule. And actually, let's look at that enzyme right now. And actually, what I'm going to do is I'm going to put a star by all the enzymes that are allosteric. So citrate synthase is allosteric. And this is citrate. And citrate is going to be consumed by an enzyme. And this enzyme is in equilibrium. And what I want to show you in this particular this reaction is that this reaction is a mechanism that you've seen before. You should have seen it in organic chemistry. It's actually a very simple mechanism. It's divided into 2 parts. The first part is an elimination. And so water is going to be eliminated. Water is going to be eliminated. And it's just a simple E2 reaction. So what I want to do is I want to draw this proton here, right? I want to draw that proton, okay? And essentially what's going to happen, and again, we're really excluding proton transfers here, but essentially there's going to be some amino acid residue that's going to deprotonate here. These electrons will kick in here, and you'll basically lose water. And again, I'm really excluding all the proton transfers, but essentially that's what happens. And what you end up generating is something called cis-aconitate. And this is an intermediate in the reaction. This is an intermediate. Right? This is an intermediate. And so it's going to look something like this. So here is cis-aconitate, right? Cis-aconitate. Notice that the carboxylates, and I'll do that in this color, the carboxylates are on the same side, right? And let me go ahead and designate this is an intermediate. So this is a cis bond. And then in the next reaction, we're going to have an addition to an alkene. In addition to an alkene, and this is still catalyzed by a aconitase. And so what's going to happen is the water is going to come in, again, water is going to come in, and essentially we're going to have an addition reaction to an alkene. So what's going to happen is water is going to hit this carbon, and this bond is going to come out and abstract a proton. So what's the net effect of aconitase? The net effect of aconitase is essentially moving the hydroxyl group from moving it from this position right here, from this position, ultimately to this position down here. And what that does is it prepares us for the next reaction. So what I've done is I've generated isocitrate, and it's still, isocitrate is still a tricarboxylic acid, or at physiological pH, a tricarboxylate. And the next reaction is something that we, and by the way, let me say this, aconitase is not an allosteric enzyme. So notice I didn't put the purple star by it. Aconitase is not allosteric, although in some ways it does regulate things. It actually regulates iron metabolism, although that's the topic of a different video. We may do a video on that someday. But aconitase is not an allosteric enzyme. And another name you might also see it written as, it's also called aconitate hydratase. Aconitate hydratase is the same thing as aconitase. Okay, the next enzyme is, or the next reaction is catalyzed by an enzyme called isocitrate dehydrogenase. And this enzyme does two things. Number one, it's going to produce NADH. It's going to produce NADH, but at the same time, it's going to produce CO2. So the carbon dioxide that's lost is this carbon dioxide. This is the carbon dioxide that's lost. And essentially, the group that's going to get dehydrogenated is this hydroxyl group. This group right there is going to get dehydrogenated, and you can guess it's going to form a ketone. It's going to form a ketone. And actually, what we're going to generate is an alpha-keto acid. We've already seen one alpha-keto acid. That was pyruvate. This is really the second alpha-keto acid we're going to see. And this is the molecule is called alpha-ketoglutarate. Alpha-ketoglutarate. So what I want to show you is I sort of flipped it upside down. This is typically the way they do it. I flipped it upside down. So essentially this carboxylate right here, I'm going to label it as 1. That's this carboxylate. If this was number 2, this would be number 2. And of course, this is the ketone that was a hydroxyl group. So we've generated alpha-ketoglutarate, and this is catalyzed by the enzyme isocitrate dehydrogenase. And we looked at the allosteric regulation of it in a previous video, so you know that isocitrate dehydrogenase is an allosteric enzyme, right? And we generate alpha-ketoglutarate. Now, alpha-ketoglutarate really has a life of its own, and it can, and we'll look at this in later videos, but alpha-ketoglutarate can actually be consumed by another enzyme, and this is actually not part of the Tca cycle, but we'll go ahead and draw it here. It can be consumed by a reversible reaction, and it's catalyzed by glutamate dehydrogenase, and what you end up forming is glutamate. You end up forming glutamate, and and this enzyme right here is also an allosteric enzyme. This is also an allosteric enzyme, glutamate dehydrogenase, right? It's also allosteric, right? Okay, but the main Tca cycle enzyme is this enzyme right here, and we're going to look at it now. And this is the alpha-ketoglutarate dehydrogenase complex. And what I want to make clear about this enzyme, number one, well, actually, let me do this. This enzyme is going to produce NADH, right? It's going to produce NADH, okay? And at the same time, you're going to put in a CoA, right? You're going to put in a CoA, and you're going to lose carbon dioxide, okay? Now, if you notice something about this enzyme, what does this look similar to? It looks similar to the pyruvate dehydrogenase complex, right? We put in a CoA, got out CO2. We put in an NAD plus and got out NADH, right? Well, it turns out that we looked at the mechanism of pyruvate dehydrogenase, and this mechanism is exactly the same. And pyruvate dehydrogenase and this enzyme are part of a class of molecules, and they're essentially called, and let me put that over here, they're called alpha-keto acid dehydrogenase complexes. They're called alpha-keto acid dehydrogenase complexes, and there's many of these. There's actually 2 others that are important. One of them is called the alpha-keto adipate dehydrogenase complex, and another one is the branched chain alpha-keto acid dehydrogenase complex. And they all have the exact same mechanism as the pyruvate dehydrogenase complex. They have the lipoic acid, they have the coenzyme A, they have the NAD, they have the FAD cofactors. They have all of the same cofactors. They have the exact same mechanism. And so in each case, you generate an NADH. So I just want to make that clear that you know the mechanism of alpha-ketoglutarate dehydrogenase. You've seen it. It's exactly the same as PDH. And when we do this reaction, we end up generating something that looks like and this is called succinyl-CoA. And this is essentially where I wanted to stop with this video. And actually, let me do this. Just like pyruvate dehydrogenase, this enzyme, alpha-ketoglutarate dehydrogenase, is also allosteric. It is also an allosteric reaction. So let me actually show you this. Let me show you this, and actually, let me do this. Let's actually put, let's put, here's phosphoenol pyruvate, right? And we get pyruvate kinase, right? And here's pyruvate, and then here's the pyruvate dehydrogenase complex, right? And we can even add this here. Pyruvate kinase was also allosteric, and so was pyruvate dehydrogenase. And this is what my biochem teacher always said. Notice, we have one allosteric, pyruvate kinase. Then we have pyruvate dehydrogenase, citrate synthase. Of course, we skip aconitase. Then we have isocitrate dehydrogenase. We have alpha-ketoglutarate dehydrogenase. We have glutamate dehydrogenase. And we also have two other allosteric enzymes that have to do with glutamate, and those are glutaminase and glutamine synthetase. But in this whole area of metabolism, we have a whole bunch of allosteric enzymes. And so this particular reaction scheme up to this point is often called the allosteric corridor. It's because we have so many allosteric enzymes that it's basically the most regulated area of biochemistry. It is extremely regulated, and a lot of the allosteric regulation occurs in the same way. And in another video, we'll actually look at the allosteric regulation, but suffice it to say for now that this particular reaction scheme, starting at phosphoenol pyruvate, is the most regulated area of biochemistry, and it's regulated allosterically. The only enzyme in this sequence, or this scheme, that's not regulated in this way is aconitase. But everything else up till succinyl-CoA is regulated allosterically, right? Is regulated allosterically. And what I want to do in the next video is pick up with succinyl-CoA. And what you'll find is that all the other enzymes in the rest of the Tca cycle are not allosteric. They are not allosteric. They're just regular Michaelis-Menten enzymes. But let's actually look at something real quick before we end this video. What I want to look at the energy yield. Do this in a bold orange. I want to look at the energy yield so far, right? So isocitrate dehydrogenase, that's our first NADH. And that NADH, as we'll see, is going to go into the respiratory chain, and it's going to get reoxidized by NADH ubiquinone oxidoreductase. And ultimately what the purpose of the respiratory chain is to pump protons that power the synthesis of ATP. Okay. And then also the alpha-ketoglutarate dehydrogenase also produced an NADH. So from the Tca cycle, we've created two NADHs up to succinyl coenzyme A, right? Also, we have waste products, and I'll do that in a gray because gray, gray, unhappy. We create a waste product, but that's going to be carbon dioxide in isocitrate dehydrogenase, and also in alpha-ketoglutarate dehydrogenase. We also produce a CO2. And in fact, what we've done, and actually let me go ahead and show you this because this is actually important. What I want to do, and I'm going to do this in a different color. I'm going to start at citrate, and let me do this in a really bright color. Notice that citrate is 6 carbons. Cis-aconitate is 6 carbons. And isocitrate is 6 carbons. Notice that as we use isocitrate dehydrogenase, we turn a 6-carbon molecule into a 5-carbon molecule. And then when we dehydrogenate even further, we generate a 4-carbon molecule. And it turns out that, that when we continue the cycle, we're going to keep moving as a 4-carbon molecule. And that's, of course, excluding the CoA. So here's 1, 2, 3, 4, right? Because there's carbons in CoA, but essentially it's just going to be a 4-carbon molecule, and as we'll find, it's the acetyl-CoA in citrate synthase that regenerates the 6-carbon molecule citrate. And actually, one thing that's important, and we'll do this in a later video, is we have something in plants called the glyoxylate cycle. And the glyoxylate cycle actually bypasses these 2 decarboxylations, because essentially, isocitrate and alpha-ketoglutarate dehydrogenases are decarboxylases ultimately. But I hope this video helped. In the next video, we'll pick up with succinyl-CoA, and we'll finish the rest of the TCA cycle. See you soon.